Welcome to section 2.3, Biological Molecules. In this section, we'll explore the molecules that make up living things: carbohydrates, lipids, proteins, and nucleic acids. We'll also see why carbon is so important to life and how even small molecular changes can have big effects. By the end of this section, you should be able to explain why carbon is central to life, describe how slight changes in amino acids can change a protein's function, identify the four major types of biological molecules, and understand their functions. Carbon is the backbone of all biological molecules because it can form four covalent bonds. This versatility lets it make chains, rings, and branched structures, creating the framework for the complex molecules of life. Macromolecules are large molecules made by linking smaller subunits called monomers into polymers. The four types, carbohydrates, lipids, proteins, and nucleic acids, each have different building blocks and functions. Carbohydrates include sugars and starches. Monosaccharides are simple sugars like glucose. Disaccharides, like sucrose, are made of two sugars. Polysaccharides, like starch, glycogen, and cellulose, are long chains. They store energy, starch in plants, glycogen in animals, and provide structural support, like cellulose in plant cell walls. Lipids are hydrophobic molecules, like fats, oils, phospholipids, and steroids. Hydrophobic means water-fearing. These molecules would rather interact with other hydrophobic molecules than with water. Most lipids are made from glycerol and fatty acids, except steroids. Lipids store energy long term. They make up cell membranes and act as signaling molecules, like hormones. Proteins serve many functions. Enzymes speed up reactions, hormones send signals, transport proteins move substances, and structural proteins give cells shape. Proteins are made from amino acids linked by peptide bonds. They have four levels of structure: primary, secondary, tertiary, and quaternary. The primary structure of a protein is simply the order of its amino acids, linked together like beads on a string. Each amino acid is joined by a peptide bond, forming a long chain. Once the amino acid chain is built, parts of it start to fold locally. This is called the secondary structure. Two common patterns form: the alpha helix, which looks like a spiral staircase, and the beta sheet, which looks like folded paper. These shapes are held in place by hydrogen bonds between the backbone atoms, not the side chains yet. The tertiary structure is the full three-dimensional shape of a single protein chain. Here, the side chains of the amino acids start to interact. Hydrophobic groups hide inside. Charged groups may attract or repel each other. Hydrogen bonds form, and sometimes strong disulfide bridges lock things in place. This is the level where the protein folds into its final working shape, ready to carry out its function. The quaternary structure is when two or more folded protein chains assemble into one big functional complex. A famous example is hemoglobin in your red blood cells, which is made of four subunits working together to carry oxygen. In sickle cell disease, one amino acid in hemoglobin changes. Valine replaces glutamic acid. This small change alters the protein's shape, causing red blood cells to form a sickle shape. These cells can block blood vessels and carry less oxygen, showing how structure determines function. Let's relate this back to the vocabulary of protein structure from the previous slide. At the primary level, Just one amino acid is different. Glutamic acid is replaced by valine at position 6 of hemoglobin's beta chain. One small swap, but it changes everything that follows. That new valine doesn't disrupt alpha helices or beta sheets directly, but it acts like a sticky puzzle piece. It still fits, but it alters how the chain folds. At the tertiary level, the problem grows. Glutamic acid is water-loving, but valine avoids water. This creates a sticky patch on the protein's surface, changing its overall three-dimensional shape. Finally, at the quaternary level, those sticky patches cause hemoglobin molecules to stick together. They form long fibers that bend red blood cells into the sickle shape. These cells break apart easily and can clog small blood vessels, leading to serious symptoms. So, one amino acid change at the primary level ripples all the way up, showing how structure and function are deeply connected. The nucleic acids, DNA and RNA, are made of nucleotides, each with a sugar, a phosphate group, and a nitrogenous base. DNA stores genetic information, and RNA helps make proteins from that information. Cells build macromolecules through dehydration synthesis, linking monomers and releasing water. And they break them down through hydrolysis, which adds water back to split bonds. Carbon's bonding ability makes life's molecular complexity possible. Small changes in amino acids can dramatically affect protein function. The four major biological molecules, carbohydrates, lipids, proteins, and nucleic acids, each have specific building blocks and functions essential for life.